Okay, so here we are going to start to discuss amino acids. And so amino acids are, of course, the building block of proteins. Um, and proteins are going to be important for many different things. So we're going to have proteins that are going to be involved in enzymes, in receptors. They're going to be important for cell-to-cell interactions. Proteins are going to be made up of polypeptides, which are linear polymers, and these are going to be made up by the 20 different amino acids that are important to us. There are about 100 amino acids in biosystems, and 20 are important for humans. And these amino acids are going to help to make our proteins that are being encoded in our DNA. So in our DNA tells our messenger RNA what type of proteins we need. That messenger RNA is going to tell which amino acids to link up to make that protein. And so the protein sequences are, of course, linked to our genetic codes. Now, amino acids are going to have both a carboxyl group and our amino group, and they are going to be attached to the same carbon. This is called the alpha carbon. Now, there are other types of amino acids, and there are the beta and the gamma amino acids. We have only alpha amino acids, so it's really easy to remember that we only have the two groups, the carboxyl and the amino group, bound to the same carbon, that first carbon, which is termed the alpha carbon. In other systems, you can have the amino group attached to the beta carbon, so this would be the alpha carbon, this would be the beta, and then here you have the alpha, the beta, and the gamma carbon, and you have the amine group attached to the gamma carbon. All right, so again, we have 20 amino acids that make up human proteins. These proteins are going to be encoded by our RNA, which is then, of course, encoded by our DNA, and they are going to make life possible. Remember, enzymes are proteins, receptors on your cells are proteins, so there's a lot of proteins in your bodies. You have neurotransmitters that are going to be made. We have here, you might as well start remembering these. We have our inhibitory and our excitatory neurotransmitter. So for inhibitory, we have something called GABA, which is basically a decarboxylated form of glutamate. And it's GABA is the aminobutyric acid. And then this, of course, excitatory is actually glutamate. So glutamate is excitatory. The decarboxylated form of that is going to be inhibitory. We also have glycine, which is an amino acid that is a inhibitory, alanine and taurine as well. And then we have aspartate, cysteine, and homocysteine, which are all going to be excitatory. Amino acids are also going to regulate. They are going to be important in signal transduction pathways, branch chain amino acids. Leucine is going to regulate a pathway called mTOR. It's got its name from cancer, the mechanistic target of rapamycin. mTOR pathway is important for a process called autophagy, which is self-eating. And so when a cell gets stressed and when it needs resources, it can actually break down itself to try to get resources in order to stay alive. And so that's a process of autophagy. And there's a signal transduction pathway that controls this. And so we have dopamine, which is going to be made from phenylalanine or tyrosine. We have epinephrine, which is going to be made from tyrosine. We have thyroxine, which is thyroid T4. We have histidine, which makes histamine. or allergic response. And so epinephrine is going to have receptors, and so we're going to have alpha and beta receptors for epinephrine. So we have alpha receptors and we have beta receptors, which we'll talk about more. But essentially, the alpha receptors are going to lead to a decrease of insulin, and our beta receptor is going to be increasing glucagon, so epinephrine's at fight or flight. And so in a fight or flight situation, you don't want to be in a fuel storage mode, which is insulin. You want to be in a fuel mobilization mode, which is glucagon. So just really quickly, amino acids and proteins. So just keep in mind what we mean by neutral when you see that name neutral. You basically do not have any charges, or the charges are going to cancel each other. In an acidic, you're going to add another acidic group to an amino acid, and in a basic, you're going to add another basic group to an amino acid. Keep in mind that they're all going to be, you're all going to have your amine group and your carboxylate group on the same alpha carbon. These are alpha, alpha, alpha. So the extra carboxylate group or the extra ammonia group are going to be secondary to the one that's the primary on that alpha carbon. So again, here we have a neutral. Here we have our acidic. And here we have our basic. So we're adding our second carboxyl group. Here we're having a neutral charge. And here we have an extra ammonia group. So amino acids all are going to have a chiral carbon except for glycine. And then they're going to have optical isomers because of this. And of course, as we mentioned before, that all of our amino acids are in L form, laboratory. Now, amino acids are also going to be referred to as amphoteric, meaning they can be an acid or a base. And when the pH is greater than the pKa, then this is going to be deprotonation. And when the pH is less than the pKa, this is where you're going to have protonation. Keep in mind that a zitteron is a neutral nucleic acid, and it's when your carboxyl donates the hydrogen to your amino group, and so you have a negative and positive charge, which makes it zitteron. And true zitterons are not really found in biological systems because, of course, your amino acid is going to be very influenced about the pH in which it's being exposed to. So the dipolar ion form of an amino acid is called a zitteron. Sorry, it's horrible. Now, an amino acid also will form a peptide bond, and we talked about peptide bonds already, but it's basically the amide. You're going to form an amide bond and you're going to lose water. So you get the peptide linkage here, and then you're going to lose the plus water. Now there are many reactions of amino acids. Amino acids are going to have the amide formations, or you can get a carboxylic acid reaction and have an ester formation. You can also have disulfide bonds form, and so here's showing a disulfide bond forming. This is epidermal growth factor, which has 53 different amino acids and 3 disulfide bonds. This is just a small portion of this molecule, of course. And so in 1986, the structure that was given a Nobel Peace Prize for determining the structure of the epidermal growth factor. But they're very large. Proteins are very large molecules. They have a lot of different amino acids, and all the properties of the different amino acids are going to have a role in how the protein actually folds and then interacts and performs its function. Now we do have essential amino acids. We have our 9 essential amino acids, and this means that basically our bodies cannot make these. We don't have the enzymes to make them, so we have to get them from outside sources. The conditional or the non-essential amino acids, we do have enzymes to make these. Some of them are called conditional because they're going to be dependent on a essential. Tyrosine is going to be dependent on the essential amino acid phenylalanine, and so that will make tyrosine essential if you have a deficiency of phenylalanine or a deficiency in the enzyme that converts phenylalanine to tyrosine, for example. Now we're going to start talking about different amino acid side chains. So the amino acid side chains are going to dictate function and reactivity. Keep in mind that a major role in the function of your amino acids is still on that alpha carbon, that carboxylic group, and that amine group on there. But then the side chains also contribute to this. Now you also have something that's referred to as a hydropathic index. And basically a hydropathic index is going to tell you which amino acids are more hydrophobic. So you can think of it as a hydrophobicity scale. So the more positive, the more hydrophobic. So when you look at these, we have our nonpolar aliphatic, and so valine, for example, I'll do the valine purple, has an HI index of about 4.2, whereas when you go to the more charged amino acids, so we'll look down here at arginine has an HI index of about negative 4.5. So the positive 4.2 is much more positive and hydrophobic than the negative 4.5. And then remember that positive, change this to black. Positive equals hydrophobic and negative equals hydrophilic. And these HI indices are in table 6.1, I believe, of your book. All right, so let's walk through these just a little bit. So we have our nonpolar aliphatic amino acids. And so these are going to be our hydrophobic. So when we start to build amino acids, the ones that get embedded into the lipid bilayer will be hydrophobic nonpolar amino acids. So they will form the portion that spans the lipid bilayer, for example. We have glycine, which is going to be the simplest. Now glycine is going to have some interaction with water because it is so simple. So it has low steric hindrance. And low steric hindrance simply refers to um, the slowing or the ability to slow down a chemical reaction due to bulkiness. I'll just call this SH, steric hindrance. So an example of this is if you have a molecule that has one carbon bound with two hydrogens and one carbon on the same molecule bound to methyl groups. There will be more steric hindrance with carbon 2 versus carbon 1 because those groups are bigger on carbon 2 and carbon 1 simply has hydrogens. So glycine has our group is essentially a hydrogen. So because of this, it has low steric hindrance, and because of this, it can often be very flexible molecule, and you're going to find it in a lot of different proteins. Your alanine, as well as your branched chain amino acids, valine, leucine, and isoleucine, are going to be more bulky, and they are going to have a lot more steric hindrance. They're going to have nonpolar side chains, and these, remember that nonpolar are all going to depend on your van der Waals forces, London dispersion forces. Mm-hmm. Okay, so now some things about your nonpolar also is that they will like to bunch together. And they tend to push, oops, they tend to keep out water and other side chains. Now we have our aromatic amino acids. And here we have phenylalanine, tyrosine, and tryptophan. These are going to contain a benzene ring or a phenyl group. And these ring structures are interesting. I want to go back just one moment. I forgot to point out proline. Proline also has a ring structure. So it is a cyclic. However, it is not a round cyclic. You can see that it is sort of a pentamer. And so this forms a kink and alpha helices. What that means is an alpha helix breaker. So if you have an alpha helix structure in your protein, you don't want proline part of that alpha helix. Now back to our other aromatics, we have phenylalanine, which has no substituents, so it's very nonpolar. And then we have tyrosine. And tyrosine is going to be one that engages in hydrogen bonding because it has the hydroxyl group. And then we have tryptophan. And tryptophan is interesting because it has a nitrogen in the endolyl ring, can do hydrogen bonding. So tryptophan is called a borderline amino acid. And it likes to hang out at the border so it can interact with both the lipid bilayer of your membrane and the aqueous environment of your cell or the extracellular membrane space. And so it's going to be found oftentimes at the interface. Okay, and then we have our aliphatic but polar amino acids, and these are uncharged. They can do hydrogen bonding with water, so they're going to be more soluble, obviously, than the nonpolars. They're going to either contain an amide or a hydroxyl group. Oops, sorry, not that one. Hydroxyl. So asparagine and glutamine are going to, the amides of the acidic amino acids, aspartate and glutamate. The hydrophobicity of these amino acids, due to that, they are found on the surface of water-soluble globular proteins. Because they have hydrophilicity, not phobicity, philicity. So they like water. Okay, now our next group are our sulfurs. So we have cysteine, which is going to be mostly undissociated and uncharged at a 7.4 pH. The cysteine can form disulfide bonds. So cysteine amino acids are not very soluble in water, but the disulfide bonds are very important in holding polypeptide chains together. Methionine is nonpolar. Sorry. So methionine is nonpolar, has a large bulky side, but what's important is it's important in the transfer of methyl groups. It cannot form disulfide bonds. And so this is a single carbon donor because it could give up that methyl group. Now our acidic and basic amino acids are going to be our charged. We have our histidine, which has the imidazole group. Oops. And histidine is going to be, of course, on the basic side. Lysine is positively charged. Epsilon amino acid, sorry, epsilon amino group. I can't remember that. So lysine has a charged epsilon amino acid. And so this is referred to as a primary, lysine is a primary amine. And then arginine has a guanidinium group. And so basic amino acids are going to form ionic bonds with our negatively charged groups. And negatively charged groups are going to be our acidic amino acids and can also be the vitamins or coenzymes. Now, our acidic amino acids, we have aspartate and glutamate. And these are going to have side chains that have our carboxylic acids. And then just keep in mind that the side chains of our negatives are weaker than the alpha carboxylic acid, or we can call this the primary. So our primary up here, call this our primary, is going to be stronger than the secondary, which are the additional carboxylic acids. And so what that means is that at a higher pKa, they have higher pKa's. So what basically this means, though, is that they negatively charged aspartic acid and glutamic acid are going to have a -1 charge. I don't want plus, I'm going to say and. Now this is going to be important for their interactions with metals, so they can interact with metals. And then they can also act as a nucleophile. A nucleophile basically will take an electron-rich group and replace a group that's attached to a carbon, so they'll be able to take on electrons. In an active site, for example, and they also are going to have ionic interactions. I'll add that site. All right, so side chains, they are going to all interact. And basically side chains can interact in 3 ways. So the first way is this A here, and they can interact as hydrophobic. And so basically, this is going to be nonpolar, and they're going to be attached by London dispersion forces. And they're going to clump together. Remember, most of our body is water, so when you have nonpolar hydrophobic interactions, their whole goal is to get away from water. So they're sort of trying to hide their hydrophobic regions. And then the second is going to be hydrogen bonds. And then hydrophilic or polar, either polar or uncharged side groups are going to have hydrophilic interactions, increasing the solubility of the protein in water. So they like to hydrogen bond to water, and they like to hydrogen bond to each other. And like their hydrophobic neighbors, they tend to react strongly and they push away nonpolar groups. And then finally, you have your ionic interactions. So to the C, or 3. And basically, you have an acid or base side chain. It leads to an ionic charge. And then that ionic charge, you have opposites attract. And so this is a very strong bond. So this is the amine of one group binding to the carboxylic of another group. And this is what is happening basically in your peptide bonds. So that's why peptide bonds are pretty strong. Now, just as a side note, histidine is going to have 3 pKa values, and it's going to have different species depending on what the pH of the system is. And keep in mind that we are up here at about 7.4. So what that means, at about 7.4, histidine only has partial. Okay, so histidine as only having a part positive charge is important for its actual function. So histidine is usually not protonated, and so it's going to be more involved in hydrogen transfer reactions. So your other basic amino acids, lysine, arginine, are going to be more involved in ionic interactions, whereas histidine doesn't really do ionic interactions because of its species at our physiologic pH is usually not ionic. And so it's going to be in a lot of our enzyme reaction sites. We'll see it a lot in there, but it's really to pick up hydrogens and move hydrogens around. And so this is just to show the different pKa's at different pHs and what species is present. And just some things to point out here is that as your pKa goes down, so your pH is decreasing. So this is at your 1.8, for example, then you're going to have all of your active sites be protonated. And on the other end, when you have the higher pH, then you're going to have them all deprotonated. And then they have species in between, partial protonation, partial deprotonation, etc. Now, pH is really going to have an effect on the side chains, and so what's important to understand is the pKa. Remember, pKa is really where the half the molecules have charged side chains. So pKa is pH, where molecules 1/2 charged. So if you have a solution that's at a pH below the pKa, then you're going to protonate your species. And if you have a solution that's above the pKa, then you're going to deprotonate your species. Okay, so just keep that in mind. So the pH is really affecting the ionization. So pH less than pKa, you get protonation. pH greater than pKa, you get deprotonation. Okay, and that is all. Most of the chemistry is now done.